Hello, this is Idris Arslan from Bülent Ecevit University, Biomedical Engineering Department and Biochemistry section. Initially, I would like to start by congratulating you due to your centennial anniversary. Recent cooperations between Ukraine and Turkey in many fields such as economical, political, and of course scientific area is very pleasant development for us. Currently, I and my research team want to contribute to this cooperation by submitting a joint research project between two countries. On this way, I would like to thank again for Ukrainian team for their project proposal. So let's get started. My presentation name is nitrogen and sulfur containing triterpene saponins and their pharmacological properties. What is saponin compound? What it mean? It means that saponins are a class of amphiphilic glycoconjugates in which hydrophilic sugars and attached to either a hydrophilic or polycyclic steroids or triterpenoid backbone. Naturally occurring products refer to not only primary but also secondary metabolites. However, research on secondary metabolites generally described ecological interactions of organisms with their environment, the pronounced biological and or pharmacological actions to great metabolite diversity and their higher tendency to interact with other biological relevant compounds. Saponins are a class of amphiphilic glycoconjugates and includes two general parts, triterpenoid or steroidal backbone and a sugar moiety. The sugar moiety is mostly composed of glucose, xylose, galactose, glucuronic acid, and sometimes rhamnose. Saponins are common compounds in nature that have been isolated from many terrestrial plants, but within the animal kingdom, they are reported only in few marine organism groups. For example, sponges, sea cucumbers, and starfish. Naturally occurring compounds are commonly found in the plant kingdom, but nitrogen and sulfur containing saponins are relatively rarely found in nature. Pharmacological properties of nitrogen triterpene saponins. The first example, N-octamin 1 inhibitory activity. N-octamin 1, a voltage gate and calcium activated chloride channel, also known as transmembrane member 16A, and function as regulation of epithelial fluid secretion, cardiac excitability, smooth muscle contraction, and nociception. The inhibition of N-octamin 1 arrests the proliferation, metastasis, and invasion of tumor cells in a human body, and also inhibition of N-octamin 1 induced or trigger the apoptosis pathway. If anoctamin 1 is overexpressed in a cell, resulting in tumorigenesis of epithelial cancers, for example, gastrointestinal stromal tumor, prostate cancer, and also lung cancer. The mode of action of this inhibition by activating of epithelial growth factor receptor and calmodulin-dependent protein kinase 2, and subsequently enhancing some protein kinase signaling, for example, mitogen-activated protein kinase signaling pathway. Actually, one of the most recently approved drugs called crofelemer, a profiled proto-cyanidin oligomer, which is extracts from bark latex of Croton leacheri, inhibits the calcium-activated chloride channel anoctamin 1. Nitrogen triterpene saponin compounds called mucoglaucosids A, B, C, D, and E, which is a 5 nitrogen containing triterpene compounds from Musenda glabra, which is a rubiaceae member. The research team discovered that both mucoglaucid ABCDE and mucoendocytes OGUPQ in human colon cell lines, results show that mucoglaucid O strongly inhibited the anoctamin with IC5 value of 22 micromolar. It can be clearly seen the molecular structure of mucoendocyte O. Another pharmacological properties of nitrogen containing triterpenes saponins is opioid receptor inhibitory activity. It's well known that opioid receptors are member of G protein coupled receptor family and also voltage gate calcium channel from a significant target for opioid regulatory action. 3 major groups, mu, kappa, and delta opioid receptors are available in the scientific world. There are evidence of a direct effects of both exogenous and endogenous mu opioids on tumor progression, recurrence, and of course metastasis in human body. The study investigating the moderate or regulatory activity of 2 nitrogen containing triterpene molecule from Musenda buteolia showed that the saponin regulated mu opioid receptor with some degree radioligand displacement. This is an interesting event and interesting results for nitrogen containing triterpene saponins.